Alright, as you can see behind me on the board, we're now going to talk about the second two phases of cellular respiration, and that's the Krebs cycle, otherwise known as the citric acid cycle, because citric acid is one of the molecules formed during the cycle, and the cycle is something that rotates. It goes around in a circle, so what you start with you end up continuously producing. And the citric acid cycle then gives way to the electron transport system, which is the ETS, or electron transport chain, as sometimes it's referred to, or ETC, and that's where the ATP in mass is made. So again, I'm going to try and keep this really simple and just show the main things that are actually occurring. So let's recap from glycolysis. We started with glucose. We split glucose down the middle into two pieces of pyruvic acid. During this process, we produced a couple of molecules of ATP by loading up a little molecule called NAD+ with a hydrogen. If there wasn't oxygen present, or there was no mitochondria, then that pyruvic acid would get converted to lactic acid and take that hydrogen back off the NADH molecule to get it back to NAD+, so it could form the ATP earlier in the system. If, however, as we're about to look at now, there is oxygen present and there are mitochondria in the cell, then the pyruvic acid will actually have a carbon cleaved off it to a 2-carbon from a 3-carbon, and have a coenzyme A molecule attached to it, making it acetyl coenzyme A. And it is this molecule, this 2-carbon molecule with the coenzyme on it, which enters into the mitochondria. Now the actual next two phases occur in two different parts of the mitochondria. The Krebs cycle takes place in the center part of the mitochondria, which is called the stroma, and is where the fluid is, so it's a water-based environment. The final part, the electron transport chain, is the enzymes are bedded into the inner membrane wall, and the hydrogen ions are pushed across and into the intermembrane space, because the mitochondria has an inner and an outer membrane. So the hydrogen ions are kept in this intermembrane space, which is just as well because it's super acidic. So the whole purpose of all of this is to produce this thing here, ATP, energy of the cell. So again, this is our mitochondria. Here's our outer membrane, here's our inner membrane, here's our intramembrane space. This stuff in this area is the stroma, which is liquid. So stroma. And so we'll look at the first phase of Krebs cycle. Now, coming in from glycolysis, we have acetyl-CoA coming through and in. Here's our cycle here, and we've taken most of the intermediaries out of it because we don't need to know them for what we're doing. We just need to know what happens. So remember, acetyl coenzyme A is a 2-carbon molecule. As it comes into the mitochondria, it binds with a 4-carbon molecule called oxaloacetate. The coenzyme A is lost and then cycles back to go and grab another pyruvic acid once it's been modified, acetyl group in the cytoplasm, and that leaves the acetyl group, the 2-carbon part, attaching to the 4-carbon part. That gives us a 6-carbon molecule, a citrate or citric acid, and it's this which then has 2 carbons removed, and that little bit of energy which is reduced every time you snap one of those bonds and remove a carbon, that's given away as carbon dioxide as a byproduct. And I've kind of drawn that line from there, which is inaccurate. It's coming down from here, but I just didn't want to bring it all the way. I guess I will, because it'll make more sense. So, that's where the carbon dioxide is coming from, and that we have to breathe out. Now, as it loses a carbon, loses a bit of energy, it reduces or adds a hydrogen onto NAD+. So that removal of the carbon dioxide gives the NAD plus just enough energy to pick up this hydrogen. We do it again, so we're now down to a 4-carbon molecule, and we get another CO2 molecule which is lost, and we get another hydrogen loaded onto an NADH taxi. And I'm just going to dot the lines up. There, and onto that one, to this thing here, and then that starts in the electron transport chain. We'll talk about that in just a minute. So we've got this 6-carbon molecule, we're cutting off carbons and releasing carbon dioxide and adding hydrogens to these little transfer molecules, NAD+. So we've done 2 of them. As we come around, we actually have a little bit of alternative energy produced, because this is guanine diphosphate, have an inorganic phosphate added to make GTP, which can act in a similar way to ATP. That's just an aside, you don't need to know that for this course. Just before we actually finish as oxaloacetate, we also load up a second taxi, and this taxi is a little bit bigger than the first taxi because we can take 2 hydrogens on this one, and that can carry around to the electron transport chain as well. A final NAD plus molecule has a hydrogen attached, and that's all of the job done. So all that happens in the Krebs cycle is to take the acetyl coenzyme A, join it to an oxaloacetate to get a 6-carbon molecule, cleave off 2 of those carbons one by one, and in so doing we load up these little taxis with hydrogens which can then be transported to the electron transport system. Out of this process we will also produce an ATP molecule. Remember, there's 2 acetyl coenzymes for every glucose, so we end up producing 2 overall because it goes through, each molecule goes through by itself. So the whole point of the Krebs cycle is to get these hydrogens transferred out on these molecules. So that takes us to the 3rd part, and this is the electron transport chain, which I've drawn up here in this area here. So I'll just put ETC in for electron transport chain. So the taxis arrive at their destination, which is the inner membrane wall where the enzymes, these little green balls here, or circles, represent. Now, the hydrogens are dropped off. Now, I will say again, because I would like you to really get this in your head, a hydrogen is that proton and an electron surrounding it. So if you take the electron away, you've just got a proton and you've got a separate electron, and that's what happens when these hydrogens are delivered. The proton, the P, is released and is pumped through the enzyme into the intramembrane space. As that requires energy, the electron here goes from a high energy position to a lower energy position, from one to the other, kind of like standing at the top of the stairs with a soccer ball and letting it go. It'll bounce 2 or 3 times before it gets to the ground. Each time it bounces, it's losing energy because it's now, the potential energy it had at the top has become kinetic energy as it moves down. So the electron starts in a high energy place here, loses a bit of energy, that's just enough energy to push that hydrogen through, and does that through the others until it is received by the oxygen. And that's what the oxygen is for. Now, if you remember from your polar covalent bonds, remember I said oxygen has a very potent electronegativity, meaning it drags electrons away from its partners and makes an imbalance in the charge because it's essentially a better magnet. Well, here it is again acting, and this is a half oxygen, it's an oxygen with a strong pull in it, and it's dragging the electron down toward it. It's the final electron acceptor. Now if that oxygen isn't present, this doesn't happen. There's nothing to pull that electron down. So this whole thing shuts down, this shuts down, you're back to glycolysis. So the hydrogens arrive, they're split into the proton and electron parts. The electrons are pulled down from one enzyme to the next toward the oxygen, and in so doing release energy, pushing the proton, which was part of the hydrogen, into this intermembrane space. So we end up with lots and lots of hydrogen ions here and very few here. So we have a concentration gradient between the inner membrane and the stroma. And at the end of this little chain, we have a specialist enzyme called an ATP synthase. And this enzyme also acts as a protein channel for protons, for hydrogen ions. So as that concentration builds up because of all of these hydrogens being dropped off and their protons being pushed through there, they flood back through the ATP synthase, and that has enough energy to fuse an inorganic phosphate molecule onto an ADP, producing lots and lots of ATP, which we can use for energy in our cell. And that's all there is to it. Thank you for listening.